Good morning to all of you, and welcome to the session for Advanced Microbiology Teaching Program in the Department of Microbiology in Mangalgiri. Today we'll be talking about rapid AST or a direct AST that is now being implemented through the guidelines by CLSI as well as by EUCAST. So we'll talk about the principle, purpose and the procedures. So the purpose, we know that we have to provide AST protocol as soon as possible for the right antimicrobials. The principle and the procedures. So what is rapid AST and why is it needed? What is the requirement? We're just talking about very briefly about the commercial methods which are available in the market and why they are successful and some of them are not successful. What do EUCAST and CLSI have to offer that we can actually practice? Do we need any special machines, any special equipments, any proprietary consumables which are required to implement these rapid AST protocols? So I will not be going into the very details about the exact method validation, but generally the concept how they have tried to validate the rapid AST protocols. So we all know that sepsis is a medical emergency and you can't afford to lose even a single hour, because every declining hour and every ongoing time the mortality increases by 8%. So we have to start the right antimicrobial at the right time, and it is not just that you start off with heavy antimicrobial treatment like broad spectrum empirical treatment just like that and start a patient with meropenem cholesterol. No, not like that. You have to have a proper targeted therapy for which you need a rapid AST protocol as early as possible. So as you can see in this case, you can see the time, as the time elapses, the patient survival rates also falls down if you don't start the right antimicrobial. More importantly is that what is the amount of blood volume that you correct. Normally what people do is they tend to will 2 ml, 3 ml, 4 ml in blood culture bottles, but the actual requirement is as high as 10 to 15 ml. At least, if not that, at least 8 to 10 ml per bottle, and at least 2 draws have to be made. 2 draws from 2 different sites always has to be sent as a paired blood culture bottle has to be sent for blood culture, and it is to be the first specimen which has to be withdrawn from a patient. Whenever you are taking multiple specimens from any patient, blood samples are taken, then the first specimen to be drawn is always a blood culture. That is standard practice, and the order of draw is also written in all the sample collection centers, all the nursing stations. That is by rule. By rule, blood culture is always the first specimen to be connected from any other patient suffering from suspected case of sepsis. And like the blood volume will also depend on the age of the patient. Again, in one of the talks I was asked that how much approximately 4 ml that you can take from a full-born neonate you can take. So this also chart is also very typical, and this is the kind of chart that should be placed in all the neonatology units also for blood culture collection. Now this is not a new thing. This problem has been there since ages, and people have tried to find some solution to have a rapid AST testing method. And this was just a few years after the Kirby and Bohr, they published their paper in this American Journal of Pathology, and this particular method was also one of the established methods for the rapid AST method, where they attempted to get the zone within 8 hours or so. That time they probably didn't even have the proper the spelling for technique also. They claimed to have 99 agreement with the gold standard. Then the gold standard, it was not even the Broth microdilution was not the gold standard that time. So there was another paper in 1973 again in the American Journal of Pathology, where they attempted to do the rapid determination of AST on urgent clinical specimens like blood cultures, and unfortunately that time they didn't have a concept of intrinsic resistance also, because they tested for ampicillin and first generation Spalosporins on Enterobacter, so they didn't even have any clue as to what the intrinsic resistances are. So the exact methodology for the Broth microdilution also did not exist at that time. It was such an old method. So Broth microdilution, whenever you're doing on the wild type strains, that also takes into account what are the intrinsic or the natural resistances which are existing in those particular strains against that particular antibiotic. So during that time there were no breakpoints, MICs which are existing over here. So Romney Humphreys, he works in CLSI, and he's the person whose team establishes the breakpoint for CLSI every year, and over the year they change also depending on the resistance development in the bacteria. We know that the sepsis cases, there are a huge number of sepsis cases worldwide, and mortality is as high as 19% if not the proper treatment is given, and it's much higher in the developing countries, and main reason being the wrong antibiotics and delay in treatment and the high prevalence of MDROs, especially in a country like NDMs. So, and MDR infections, like most of the patients, they're getting wrong treatments also. So they did a review as to what are different AST methods, commercial methods which are available in the market. They did a review and tried to correlate with the clinical outcomes. And these are the tests that they had evaluated, all different commercial tests which are available. I think this was a paper published in 2020, I think. And then all these rapid tests have got different turnaround times, like 7 hours, 3 to 5 hours. And most of them are CE-IVD approved and US FDA approved also. And they found that this one of the papers published in 2020, this was a multi-centric trial, and they performed Malditov as the standard of control, that is the SOC, the standard of control, and they used that method. This was actually accelerated pheno system was used, and it was compared with Malditov system. This was a 2-arm randomized control trial. They correlated with the antimicrobial stewardship practices also. There was audit and feedback by the ID pharmacist also, and they found that it has decreased the turnaround time to optimal therapy. TOT is turnaround time to optimal therapy. There were no differences in the mortality. There was no differences in the length of stay or adverse event, but definitely it did improve the timing in which the right antibiotic was started. So I just discussed one of the commercial methods that was published in, I think, Nature. In this method, what they do is that they have got this blood culture bottle which has come out positive. They mix it with an agarose and then make an agarose mixture, and that agarose mixture is added on these microtiter plates. Now these microtiter plates are slightly different. They are actually two-chambered. One has got this is a micro patterned chamber where they put the agarose, and this other one has got the freeze-dried antibiotic well is there. So as you can see, there are two wells over here, one larger one and smaller one. And then after that, they add the agarose mixture is added over here, and then they later on they add the culture medium to it, and then they incubate it, and they focus it under a microscope. So that is one of the commercial methods that's available. But then again, it requires special microscopes for viewing. So considering the existing the logistics in most of the labs, these kind of tests, they are good, but they are not, all of them are not possible to perform. So again, this was a review that was published in Nature again, which has got numerous number of commercial methods which are available. Some of them are in different countries, some of them are US FDA approved, some are CE-IVD approved. So, but then they all require different or special equipment are required for the interpretation. So none of them are normally available freely in the market, because most of them, they require validation through the reference method, that is the Broth microdilution as per ISO 20776. They may not compare with the Broth microdilution. Most of the positive blood culture samples are monomicrobial. So we can always use that particular principle and incorporate directly on the AST rather than going for high-end machines. Other than that, CLSI makes a lot of amendments in their the guidelines, and those amendments cannot be extrapolated onto these commercial systems. So before we actually get on to the rapid AST, there are certain terms that we need to understand, like what is the MIC, what is MDC, what is breakpoint, what is SIR. So I think most of them you are already aware of, but I'll just go through basic things. That what is breakpoint? Breakpoint is nothing but the concentration of an antimicrobial that separates two organisms, or you can say populations of microorganisms. That's a definition given in Victor Lorien. Now there can be more than one breakpoint that can exist for a specific antimicrobial or a microorganism combination. Now these are the different ones, like epidemiological cutoffs are there and clinical cutoffs are there. Basically MIC, it's a pharmacodynamic parameter, and it keeps on changing over the time depending on what is the resistance mechanism that is developing over time, and then these are the ones which are changed by CLSI or EUCAST, and they vary from country to country. and from official body depending on what is the concentration of antibiotic against the bacteria is tested. So these are, as I said, they are values which separate populations of organisms and can be presented as MIC or zone diameters. We need them primarily for clinical interpretation to tell the clinician whether what antimicrobial can be given, what cannot be given, basically. So there are some facts and myths. What we call about the MIC is that this is the highest dilution or the lowest concentration which shows no visible growth. That means we cannot see 10 to the power 4 CFU per ml, maybe we can see 10 to the power 5, 10 to the power 6, but then if there is no visible growth, that does not mean there is no growth. No visible growth does not mean no growth. So it's not a true value because it's impossible to determine the actual MIC. Reason being that whenever we are doing the serial dilutions or double dilutions, we are checking for, let's say, 2, 4, 8, 16. So if you are looking at this, at 16 there is no growth, but at 8 there is growth, we will label the MIC as 16. But what about 15, what about 14, what about 13? So there could be growth, there may not be growth. So though that value between 9 to 15, we cannot determine that true value. So at the most we can do geometric dilutions, but that's again a very, very tedious process, and that cannot be done in routine labs, can be done only in the referral labs only. Now MIC does not represent the in vivo concentration. In vivo concentration will happen with whatever amount of drug that is given. Now that is again a very tedious task to decide what should be the drug level versus the MIC level, because the anatomical therapeutic classification ATC, defined daily dosing, it is done by checking for therapeutic index, that is therapeutic safety of the drug, what is the maximum amount of drug that can be tolerated by a healthy human, what are the trough levels which are reached in the blood after a standard dose, and whether that level be effective in vivo or not. So that's a long protocol to develop those levels. So it does not represent the in vivo concentration. So if a MIC is 2, despite being susceptible, the bacteria may die, it may not be killed depending on what. So it does not represent an in vivo concentration. So basically it is a phenotypic endpoint. This is a very clear-cut definition. It's a phenotypic endpoint in a defined and a standardized but a highly relative system. So what we are testing it in the lab is highly defined, but we are actually practicing in a highly relative system, that is the human body. So MIC is measured in vitro, but it is not really what actually happens in vivo. But as of today, MIC is the only standard measure of activity of a drug against a particular bug. So as you can see that this is the Broth macro dilution method, where you can see the tubes are there at 0.25 concentration, 0.5, 1 concentration, there is growth, but at 2 there is no visible growth. So that 2, there could be a growth. So once you start doing subcultures of these tubes, we'll find that there is growth over here. When we see the subculture of this thing, we find there was growth which could not see. So there is growth coming once you make a subculture. Once you make a subculture from 4, we see a growth. So that is the large concentration where there is no growth after subculture. That is the actual killing of the bacteria, or the bacteria has totally got killed, and this is the minimum bactericidal concentration. And by convention, it is said that the greater the difference between the MBC and MIC, if the MBC is very, very high and the MIC is low, then it is usually a statin drug. If the MBC and MIC difference is slightly low, then it is usually considered to be a sidal antibiotic. But then that's again a very relative definition, because that keeps on changing depending on what are the in vivo conditions. So as we know that the referral labs, which are CLSI and the EUCAST, these are two main labs which are developing the breakpoints, and there are reasons why we need to stock our pan susceptible isolates, because they are the ones which are used to demonstrate develop the epidemiological cutoffs. So what are the epidemiological cutoffs actually? Whenever we have got two types of bacteria, one of the wild type and a non-wild type bacteria. The wild types are the ones which do not have any discernible resistance mechanism, and they are likely to respond to a particular antibiotic. And then the other ones are the non-wild types. That means they are usually taken from the patients, and they are likely to harbor some resistance mechanisms. So basically, wild type strains, there can be mutation leading to altered target sites, acquisition of the foreign DNA, upregulation of these are different resistance mechanisms. Once the wild type strain acquired, then they become the non-wild type. So basically, we are checking for the antibiotic concentrations in the wild type, but that's why we don't know whether this MIC will be result in clinical treatment or not. So normally the epidemiological cutoff MICs are usually lower than the non-wild type MICs. So CLSI normally uses 100 unique strains for at least 3 different labs. They are all calculating the MICs from the wild strains to develop the epidemiological cutoffs. So these are basically the genetically and phenotypically stable strains which can be used as quality control strains with stable QC-MIC ranges, that is the ATC strains. So normally ATC strains are the genetically stable wild type strains which do not have shifting MICs once we unless we are not able to maintain the proper stock mechanisms. So clinical cutoffs are basically the MICs that separate the strains where there is a high likelihood of treatment success from those bacteria where the treatment is most likely to fail. So the clinical cutoffs are slightly higher than the epidemiological cutoffs, and they are derived from MICs of pathogens isolate from prospective human clinical studies who have been given the usual prescribed dose. For example, ampicillin is given as 500 mg TDS. So 500 mg TDS will produce a certain level of concentration in the blood. So against that the MIC is calculated in vivo and in vitro. So what we are seeing in the clinical cutoffs is whether at this particular MIC there will be clinical response or a microbiological response or not. So normally in these MIC ranges we will have a clinical response and the patient is likely to recover. Now this is slightly outside the scope, but still I'll just go through PK/PD cutoffs are like, for example, I told you what is a therapeutic index. Therapeutic index is basically the safety parameter of an antibiotic. Like for example, if I say that piperacillin tazobactam, the usual dose is let's say 2 gram every 8 hourly can be given. But at the same time, if it has got a higher therapeutic, it's got a good therapeutic index of safety, in that case you can increase the dose. So at let's say let's say at 2 milligram 8 hourly, if the MIC that was determined by CLSI was let's say around 21 millimeters, or maybe like around 16/4 microgram per ml. So in that case, at that it will respond. But what if the MIC rises to 32 and 8? That's for peptazo, 32 and 8 rises above that. Then can you increase the concentration or you can increase the dose of the antibiotic? So once the increased dose gets validated against the increased MIC, that becomes the PK/PD MIC basically, or the PK/PD cutoff. So as you can see that whenever we place a disc on the Nagar plate, there is a logarithmic fall in the MIC as we go away from the edge of the disc. So that's a very typical thing. So higher the concentration over here, then the larger is going to the zone. At the same time, if you have, I'm just trying to just tell you this concept that if you have got different concentrations of the same antibiotic, it will produce smaller zones. So at the concentration there is no zone, that will become your MIC. So once you extrapolate it onto a strip, that becomes your MIC strip only. So basically they are all small discs placed together, and that produces your elliptical zone. Now introduction to the rapid AST from blood culture method. So the main purpose is for the early targeted therapy for sepsis. It is normally done by distribution method. The standard breakpoint zones we take for 20 hours, they cannot be extrapolated over here. So like for example, if you have a standard zone of let us say 25 millimeters of piperacillin tazobactam at 22 hours in CLSI, you cannot use that same zone for 4 hours or 6 hours for peptazo and E. coli. So that means you cannot extrapolate whatever is there which is already established. So we have to have a separate, we have to develop separate breakpoints, and we have to establish the proper epidemiological cutoffs at 4 hours or 6 hours or 8 hours. But the only issue is that with short incubation, the catch is there, that with short incubation, there are smaller zones in the wild type strains and the larger zones in the non-wild type strains. Now we would imagine that because wild type strains do not have an intrinsic resistance mechanism, they would have larger zones at short incubation, and with the non-wild type strains which have got some resistance mechanism, they would have smaller zones at short incubation, but it is the opposite. So that's one of the things that has led us to the fact that we cannot extrapolate these zones to 4 hours, 6 hours or 8 hours. So that is one of the reasons why we cannot extrapolate that. So that is why we need to develop a newer system or newer methodology for a rapid AST method. So both EUCAST and CLSI have developed their own methods. Both the principle is the same. Principle is widely the same, though there is not much difference. Main differences in the incubation time. I'll just tell you what the differences are later on. So EUCAST developed this particular method, the EUCAST rapid disk diffusion method for antimicrobial susceptibility testing directly from blood culture bottles. That was again by this is the original publication of EUCAST in that was a method validation protocol in 2019, published in JAC. By Gunnar Kalmeter is the main scientist who's involved in the European EUCAST guidelines development, and CLSI they published this paper in 2018, but till recently they did not come with the full-fledged method. So this is mainly published by Romney Humphreys, who's a person who makes his team makes the CLSI breakpoints. So that's the British European guidelines. This is the American guidelines. By far, mostly they are very similar or parallel. The principle is the same. Now, before going into details, how do we represent breakpoints? Now, this is one of the methods of representing breakpoints. On the x-axis, we have the zone diameters. On the y-axis, we have the MICs. Now, greater the lesser the MIC, the wider is the zone diameter, and vice versa. The smaller the zone diameter, the higher the MIC. So that is the basic principle. But there are many outliers at times. Like outliers are like if you got like this one. This one is got a zone diameter small and the MIC is also less. So ideally this should have been over here with a smaller MIC, but it is falling over there. So that's a possibility that the disc is not working or the antibiotic is degraded for whatever reason. That is one of the outliers. That's a major error. Very major error is when, despite a very high MIC, there's a big zone size. So that is a very major error. Ones which are in the between. So this is where our susceptible or intermediate or resistant that lies. But before that, there's a thing known as the area of technical uncertainty that is defined by the EUCAST. Now, what is area of technical uncertainty? So this is a scatter plot method usually used by CLSI. This is the histogram method which is usually used by EUCAST. So as I can say that larger the breakpoint zone, lesser the breakpoint MIC, right? And this is CLSI, this is EUCAST. So what is the area of technical university? Is basically area of technical university includes the technical errors. For example, there are variations in the media, variations in the pH, variations in the incubatory conditions, variations in the there are numerous variables, the temperature, the time of incubation, the interpretation. So this is the area of technical uncertainty where you cannot do the interpretation. You cannot say if it is this, it is intermediate. No, first you have to rule out all these things. Once you rule out all these things, then you can call it interpretation as intermediate, or we call it the susceptible increased exposure. So this is what is known as the SDD also at times. When this gets validated, now EUCAST says that this is, as per EUCAST, this is not a valid thing. EUCAST says that, okay fine, you increase the dose and duration, the patient is likely to respond with this MIC. But with CLSI, they have actual validated clinical studies which say that, okay, if I am using 4.5 gram of piperacillin tazobactam with an MIC of 32/4, in that case, it will go to respond. So same thing goes for other. As you can see that most of the CLSI protocols, the SDD has been defined for the cephalosporins or the beta-lactam drugs because they've got a very good therapeutic index. That means their safety is very high. So you can afford to increase the dose and duration in that case. So SDD are usually defined for the beta-lactam antibiotics. So that is how the breakpoints are being represented either as scatter plots or as histograms. Now in the histogram, what is happening? Again, they are checking for the number of readings. This is the number of readings they are taken for a particular isolate, and this is the inhibition zone, and these are the MIC. So there are three parameters which are checked on the histograms, and the ones over here which have got the high zone diameter, which have got a lower MICs over here, and the ones which have got a small zone diameter have got higher MICs. So something that is in between, which we cannot determine whether to label it as intermediate or not in the initial phase is the area of technical uncertainty. Once we get all these errors erased, then we can consider that as intermediate or susceptible increased exposure. Now, how do we arrive at breakpoint zones? Now, this was I think this was taken by Dr. Dev also earlier, but I'll just revise this part. Now we know that the Broth microdilution is the international reference method. Now that is based on the ISO 20776 2019. That's the reference standard which uses a 0.5 McFarlane turbidity, right? Now EUCAST and CLSI, they set the clinical breakpoints for the wild type and the non-wild type strain. So first they see the wild type strain, whether I can test for cholesterol and Proteus or not. So they take wild type strains of Proteus and then they check for cholesterol and they find out that it is not responding at all. So there is some intrinsic resistance present in Proteus against cholesterol. So we won't use this antibiotic at all. So wild type strains are also defining the very baseline level, the most baseline level, the very basic intrinsic nature of a bacteria without the presence of any acquired resistance mechanism. What is going to be the reaction of the bacteria against a particular antibiotic? Like for example, if you try to give cefotaxime in Pseudomonas aeruginosa, it won't work because there is an intrinsic AMC-beta-lactamase that is going to degrade that cefotaxime. So wild type strains will determine the very basic baseline level as to at what concentration a strain with no discernible resistance mechanism will have the cutoff MICs. These are the epidemiological cutoffs. Once they are established, then they take known resistance mechanisms. Strains with known resistance mechanisms are taken from, and that even they have a resistance mechanism, it should be for some other class of drug. Like for example, you can take a known NDM strain and then test it for aminoglycosides, that's fine. But if you take a known NDM strain and try to test it against cefotaxime, that's stupidity. Right? So you have to test with even there is a known resistance mechanism, so accordingly you have to choose a drug. For example, if you have a drug with known OXA48 or known KPC, in that case, I'll check for Cipta-AV or maybe I'll check for Ceftriaxone, Tazobactam, I'll check. But so that is the basic principle of how to choose your non-wild type strain. It should not be ideally against the same class of drug. Or if you are checking, like it should be higher. Like if it is cefotaxime resistant, you can afford to check for a higher generation cephalosporin, but not a lower one. So that is the principle of the BMD reference method. Then what happens is, once we make these scatter plots or these histograms, then we extrapolate it to the breakpoint zone. So in fact, this MIC, this is a zone, this MIC, this is a zone. So that's how accordingly the zones are determined. So using the same strains and using the BMD as a metric standard. Now, once you have used this method, that becomes your basic scale. That is the basic term of reference. That is the metric standard against which your Broth your breakpoint zones are normally established. So a range is set against the MIC values, and areas with technical university, they get extrapolated to gray zones like intermediate, SDD or SIE. So once you rule out the errors, you rule out the media is standardized, the incubatory condition is standardized, the technical competence is also standardized, everything is there. Once it's standardized, you know that this strain is showing an MIC of, let's say, 16 by 24 at this particular against this constant, this particular antibiotic. So we'll have to put it in the I category only and not in the S or the R category. And these are set at 35 degrees at 18 plus minus 2 hours, and that's how we get the clinically interpretable breakpoint zones which we use for daily AST reporting. So how did they arrive at the rapid AST breakpoint zones? Now what is happening is, this is a very interesting thing. Can we extrapolate the distribution based zones to rapid AST method? The answer is no, because I said earlier also that what we are testing in the routine distribution method is basically a colony based method. Using colony based method, we are picking up the colony, we are making an inoculum of 0.5 McFarlane turbidity standard, we are creating an inoculum. Now what is happening in the rapid AST method, we are taking the positive blood culture bottle directly and using it as an inoculum. Now in that case, it is not the same as this. So you cannot compare both the methods together. So that is why we have to create those conditions. So that is why in these rapid methods, we have to create the inoculum by spiking the blood culture bottles and using them as positive blood culture bottles. So we are using positive blood culture bottles as an inoculum and not the colony as an inoculum. That is where the catch lies, and that is why this entire method validation was such a long process, and that's why EUCAST and CLSI, they took their own time to make this entire protocol. This is a very, very tedious process. So what they did was, like, Broth microdilution is a reference method that is colony based. We all know that, discussed that, based on ISO 20776. We set the clinical breakpoint for the non-wild type strains. Then in this, what we have done is they spike the blood culture bottles and they make the inoculum. So they may take a suspension around 100-200 CFU per ml. They spike the blood culture bottle. Now this particular paper does not mention the exact volume, how many 1 ml, 2 ml, or how much ml they are taking. Are they taking 8 to 10 ml? They are using that amount to spike the sterile blood culture bottles and then adding around 5 ml of defibrinated horse blood in that bottle and then incubating it, and an average time of 8 hours to 12 hours, approximately 11 hours they are taking. Average time, the bottles are coming as positive and they are processed within 0 to 14 hours after the signal turns to be positive. Now they are simulating the entire conditions as to what happens in the lab. Like for example, if you got a 24/7 lab and the blood culture comes positive, let us say at 3 AM, right? And the technician is sleeping over there and he wakes up around 6:30, 7:30, 6:30 or 7 o'clock. He wakes up, he sees that it has flashed positive at 3 AM. So there is a elapse of 4 hours. So that, to take that account, so they have kept a range of 0 to 14 hours after signal comes to be positive. So that is a normal time when we are likely to process these bottles during that time. And multiple, they have, before that, in the pre-analytical step, they have done the multiple studies that after the signal comes positive, the bottle within this time only remains in that range of somewhere around, like 0.5 to 0.6 McFarlane turbidity standard, remains over there only, does not increase after that. Then after that they develop the rapid AST DD protocol. Now they use this as the inoculum. Once this spike blood culture pulse comes to be positive, then they use that inoculum, take around 100 to 150 microliter. The volume is around 125 microliter is taken, and then that is put on the molaret and agar. It is done, the swabbing is done, and then the same antibiotic discs which are normally used, they are the ones which are used to put on these plates. The incubation is done for around 35 degrees and read at 4 hours, 6 hours, 8 hours, and finally at 16 and 20 hours. So there are total 5 readings which are taken. All the readings are taken with the lid to be open, and after, if you're taking the reading at 4 hours, so within 10 minutes of taking the reading, you have to put it back in incubation. Within 10 minutes only. You're taking at 6 hours, then within 10 minutes you have to take the reading immediately, and you have to take the reading. It's not interpretation. Just mind it's the reading, not interpretation. So you have to take the reading and then put it back in the incubator, and finally at 16-24 hours, 20 hours. So the purpose of this entire exercise is to simulate a positive blood culture bottle and not the routine disc diffusion methods. That's why this entire process is going on. So this is what EUCAST has done, is that EUCAST has made these species which are available, for which breakpoints are available for E. coli at 4, 6, 8 and 16 to 20 hours. Klebsiella pneumoniae, so there is a huge drop down menu which is there, and all these different breakpoints have been established at 4 hours, 6 hours, 8 hours. For example, if you got an E. coli, you've got let's say you have done the rapid AST and you are not able to read at 4 hours, for example. For some reason you are not able to read at 4 hours, your technician is not there, you can read at 8 hours. So there are 6 hours and 8 hours also. So they established clinical breakpoint for rapid AST in all different timings for these group of bugs by EUCAST. So this is the EUCAST rapid AST method which is validated for these bugs against these timings. So as you can see, Pseudomonas you cannot read at 4 hours. At the same time, Acinetobacter has not been established at 16-20 hours. Same goes for Enterococcus faecalis and Enterococcus faecium also. Some are established, some are not established. The reason is for maybe the bacterial growth is different, or the bacteria responds differently on the medium in all these cases. Now this is a proportion of the readable zones. This is a readable zone, not interpretable zones. You see, these are readable zones. They are possible. They are not possible to interpret. Okay, so you can read E. coli at 90% strains. You can read at 4 hours. 96% Klebsiella you can read at 4 hours. 99% Acinetobacter you can read at 4 hours. So 55% only at this thing, for Staph aureus. So this is the what percentage of the strains that you can read at 4 hours. If you can read at E. coli at 4 hours, fine, then do the reporting. The breakpoints are established for rapid AST at 4 hours, you can give the report immediately. Same thing, 99% strains or 83% strains, they can be read at 6 hours. So at 6 hours also, if you can send the report, there are established breakpoints for that also. Similarly for 8 hours and 16 to 20 hours also. So at 20 hours, you can see all the strains can be read except for these two, they can be read. And by 8 hours or 6 hours, most of the things you can read. So you can give the AST report, rapid AST report within 6 hours to 8 hours. So they are so careful in establishing the breakpoints that breakpoints are different at 6 hours, breakpoints are different at 8 hours also. This is just a 2 hours difference, they can be a huge difference also. So you cannot extrapolate, okay, if I am reading at 8 hours, so let me use 6 hours breakpoints. No, the 6 hours breakpoints are different, 8 hours are different. How many isolates to test? So what EUCAST has done is that they have tested for, let's say, 59 isolates for piptazo, and they have done the duplicate readings. So 59 into 2 is 118. Similarly, 54 readings for ceftolozane, tazobactam, 108 readings. Two readings have been taken for each isolate. So CLSI mentions 100 isolates, but they have taken more than 30 isolates, so maybe 59, 60 isolates they have taken. What is the basis of using these number of isolates? I don't know. But then at least what roughly say that it has to be more than 30 non-repeat isolates should be there. Each tested twice. So this is the E. coli and piptazo EUCAST distribution at 16 hours versus the broth microdilution at 16 to 20 hours. So this is the validation. So you can see clear cut that this is where the cutoff is. This is the breakpoint zone. Like around say 19 millimeters is the breakpoint zone at 16 to 20 hours for E. coli in piptazo using the broth microdilution method. So what they do is, this is again E. coli and piptazo. This now what they've done is when the blood culture is spiked with E. coli coming out as positive, let us say 10 hours, they put the rapid AST. So rapid AST is put, that is one method. That same method is validated against the broth microdilution, that is the reference method. So they have made the different histograms at 4 hours, 6 hours, 8 hours, 16 hours and 20 hours. So on the basis of this, they are establishing the breakpoint zones at different times for different bacteria against different antibiotics. So that is the kind of precision and that is the kind of thought has got into the development of the rapid AST protocols. Similarly, this is for CLSI. CLSI, like if you can see that in EUCAST, there are specific bacterial names are there, like E. coli or Klebsiella or like that. In this case, it is generally for all enterobacterials and for Pseudomonas. There are no involvement of any gram positives over here. They say that it's not to flash. If it flashes within 8 hours of flashing, you are supposed to process the bottle. They have got a specific test procedure. Invert the bottle 5 to 10 times. 20 gauge needle to be used. 4 drops of the broth to be used. Lid jar to be kept open for 3 to 5 minutes, but not more than 15 minutes. Within 15 minutes, you put the discs and you put for incubation, and to read at 8 to 10 hours or at 16 to 18 hours. So that's the difference that they have. Instead of using 4 hours, 6 hours, 8 hours, they are just using 8 hours, 8 to 10 hours and 16 to 18 hours that they are using. And they have got a interpretation tables given in the M100 document under table 3E2 and table 3E3 for Pseudomonas and Enterobacteriaceae. So as you can see that these are the breakpoints for 8 to 10 hours and this is the timing that is mentioned over here. 8 to 10 hours there are no breakpoint zone for ampicillin and enterobacterials, but for 16 to 18 hours there are present. Similarly for this also, for cotrimoxazole also at 8 to 10 hours there are no breakpoint zones. So these are the, at 8 hours you can interpret only ceftriaxone, ceftazidime, aztreonam, tobramycin. That's it. At around 8 to 10 hours they are validated for these 4 antimicrobials, but at 16 to 18 hours all of them are there. So that is one issue with enterobacterials, that you can give the interpretation only for these 4 antimicrobials: ceftriaxone, ceftriaxone, ceftazidime, aztreonam, tobramycin and cotrimoxazole at 16 to 18 hours. Otherwise you can't give for earlier ones. Similarly, it goes for Pseudomonas aeruginosa also. Pseudomonas aeruginosa, you can see there are no breakpoint zones at 8 hours, 10 hours, 8, 10. There are no breakpoint zones for ceftazidime and meropenem, whereas for tobramycin and cipro they are there. So if we are given a choice, then I would say that EUCAST is slightly a way better because it has got a very, they have validated the breakpoints for most of the antimicrobials at different times. But the only problem with EUCAST is that since it is species specific, we need to have machines like Maldives, right? Because Maldives do the immediate identification, and with that immediate ID at 4 hours, you can give the report at 4 hours also. So the purpose of the EUCAST was that method should be valid for relevant bloodstream infection pathogens and agents. The time of the result should be shorter than the standard workday. Material should be available in a standard microbiology laboratory, except for the MALDI. That is one problem. That otherwise most of the things are available. The method should be easy to perform and control. It should be independent of the blood culture system. I mean, it should not be like it is only for BD Phoenix or it is only BD or it is only for Bactrilide. So it should not be. It should be independent of the system, and it should be freely available on the EUCAST website. So they have tried to follow all these things. And in April 12, 2022, EUCAST released the official EUCAST rapid AST method directly from blood culture bottles. The essential steps are, like I told you, the specific volume I have mentioned, using 125 microliter of the positive blood culture broth, which is processed within 0 to 14 hours, unlike that for 8 hours for CLSI. There is no centrifugation which is required, no dilution, there is no alcohol is required. You take the positive blood culture bottle and straight away put it around 125 microliter, you put on the plate, incubate and read at 4 hours, 6 hours, 8 hours, 35 degrees plus minus 2. And since April, now in April it was possible, like what happens if there is no growth at 4 hours, no growth at 6 hours, what do? Can you interpret 16 hours or 20 hours? Yes. So after April, they incorporated the breakpoint zones to be able to be read at 16 hours. For example, if your plate is 8 hours, you forgot to read. Now what do? Do I extrapolate the time or increase the time? So they validated that it is possible to prolong incubation to 16 hours but not beyond. So these direct AST plates can be read as routine AST plates, which we normally read at 20 hours. So that can also be done. Breakpoints are specific for each species and each reading time. So we have to be very careful. You have to have a long, huge chart is there for these breakpoint zones, because every time has got a different breakpoint zones, and MALDI is needed for that because you need to have the accurate identification. So EUCAST, like I said, is validated for E. coli, Klebsiella, Pseudomonas, aeruginosa, and these four ones, they are normally not present in the CLSI. Only Enterobacrils and Pseudomonas is there in CLSI. EUCAST method is species specific. CLSI is for Pseudomonas and Enterobacrils in general. There are different incubation times. Like for example, for EUCAST it's 4 hours, 6 hours, 8 hours, and for CLSI it's only 8 to 10 hours. There are no gram-positive organisms present in the CLSI direct AST method, unfortunately. But in the EUCAST, there are gram-positive there. Again, the volumes is again very specific, 125 microliter versus 4 drops from a 20 gauge needle. Positive signal has to be processed within 0 to 14 hours actually, is a mistake, versus 0 to 8 hours in CLSI. So that is these are the main differences against the CLSI versus the EUCAST rapid AST methods. Now the question arises, then when we do not have automation. Now can you still implement the principles and practices of antimicrobial stewardship when we don't have automation? This we all have witnessed. I have said this thing again and again, that what happens in the government hospitals, we get manual blood culture bottles during off-duty hours, there are no technician available or there is no resident available. We try to do the manual processing, and we send the blood culture bottle, let us say, send to the lab at 7 p.m. but the lab is closed at 4 p.m. and samples are not accepted at 3 p.m. That is what I have witnessed in many of the government medical colleges. This happens. Samples arrive the next day on Friday at 10 a.m. and gets incubated. By Saturday, blind subcultures are made. This is what we are practicing right now. Also, we are trying to observe the headspace stability. Headspace stability is one of the most critical things that has to be observed in all blood culture bottles when you are processing them manually. If you see headspace stability, please make a gram stain. This gram stain is not made routinely, and on Sunday if there is a growth, it's a holiday, nobody is coming there to process it, and ultimately Monday a manual IDST is done. Pediatrician is still not informed. Again, non-communication with the clinician is a crime. You have to call the clinician, talk to them, speak to them, message them, whatever it is possible. You have to tell the clinician what are the critical alerts. For that, you need to define your critical alerts. So, you have to accept the sample irrespective of whether, because blood sepsis is an emergency, you cannot just refuse the sample. You have to accept the sample and advise for other tests like PCT or CRP from emergency labs. You have to notice the headspace turbidity and always make a gram stain, and in the gram stain you find a gram negative, inform the clinician as early as possible and try to put a manual or a direct AST. Now it may not be exactly the same as what is happening over here, but at least you can get a rough idea. Yes, in this case cefotaxime is not to be given, it is definitely an ESBL. So you have to first identify what are the critical alerts. We have identified the critical alerts in our department, like blood culture bottle if it is flashing positive. You have to call a group immediately if the gram negative bacilli is there, gram positive bacilli is there. Now what happens if you have a BHI bottle which is positive? Let us say you got a GMB, you can put a direct AST from blood culture broth. That is the EUCAST guidelines are existing, right? If there is GPC cluster, you say okay, it's coagulase negative stab. But what we have learned in our pure culture exercises in our MD during our MD exam, that you can perform a tube coagulase with your broth also. If you get a positive blood culture bottle, you get GPC in clusters. You can take that, you can centrifuge it, you can take the supernatant, you can do a tube coagulase even with that positive broth. And when you make a subculture on blood agar, always put up a vancomycin disc. Reason being that many times there are co-infections, not co-infections, actually gram-negative infection, but then since the cubital fossa is not cleaned properly, there is growth of coagulase negative staph along with that. To prevent that staph in that inoculum, the vancomycin disc is there, and around that vancomycin disc you will see the growth of gram negatives. So from where you can harness, you can retrieve those gram negatives, because the entire plate will be flooded by coagulase negative staph with few occasional colonies of gram negatives. That will be the true pathogen. So always try to put up a vancomycin disc on the primary inoculum. If you see gram positive cocaine chains in your positive blood culture bottle, you can always put a biologically agar with 29212 as a control strain and inform the clinician by SMS, WhatsApp or call them. So always going for a direct AST. Like I said, the mortality always increases by 8% if you don't process the sample properly. So the therapy has to be early, the therapy has to be targeted. You have to define the proper posology of the particular antibiotic for that particular patient, because you have to tell them that if it's a beta-lactam drug, how is it to be given? If it is an aminoglycoside drug, how is it to be given? Hand hygiene and hospital infection control practice have to be strictly followed. EUCAST and CLSI guidelines can be applied to the lab. So in CLSI and EUCAST you can apply. With EUCAST and now with the recent introduction of the AMSP NAP AMR module also in the state, now it has been made mandatory to acquire the MALDI-TOF also in every institute. So it's an absolute essentiality now. It's a basic machine now. It's not a essential. It's not a privilege for us. It's an essential commodity now. So the take-home message is that you need a 24/7 lab working 365 days a year with proper diagnostic lab and manpower and competent signatory authority. Just keeping an automated blood culture machine bedside will not suffice. You have to process it as early as possible. Clinical reporting, like in JIPPER also, clinical reporting starts at 7 AM in the morning, so that by 9 o'clock all the reports are ready and all the reports are sent through WhatsApp or through SMS. All the PDFs are sent to the clinicians by the time they reach the rounds. So you need an active clinical microbiologist required, and this can be done in the absence of automation. Even if you don't have an automation, still you can go in for active antimicrobial stewardship, like I mentioned in the previous example of in case of manual samples. So do not wait, please innovate. Try to incorporate all the principles and practices of clinical microbiology that we have practiced during our MD program and in the MD teaching, and try to decrease the turnaround time of such cases. Thank you so much.